The next platform is the Illumina or Solexa Genome Analyzer. So for this platform, they do not use emulsion PCR, but instead they are using bridge PCR method to generate the fragments. So for this method, all the fragments which are anchored with the adapters are attached onto a solid surface where they remain immobilized and clustered. So from here, they will perform the PCR. So you can see here the shape is like a bridge. That's why they have the name which is called bridge PCR. So in any one location, one type of fragments would form a colonies. So we call this as polonies, PCR colonies. So in this location, you have one PCR colonies, which is the same fragment, while another position you have the DNA fragment, another DNA fragments which form a colony in another position on an array on a chip. So how do they perform sequencing? It is different from the 454 platform where they are deposited into a well. So in this case, all the fragments which is adapted on the surface on the chip, they will be using a 4-color cyclic reversible termination. So for this Illumina Solexa platform, they are using reversible termination method, where at any one reaction, they will incorporate with a fluorescently labeled nucleotide. So if let's say the template is G and the fluorescent label C would be attached. So for different fragments, all of them will have nucleotides being incorporated at the same time, which is quite different from 454, where some of them are faster, some of them are slower. So for this Illumina, all of the reactions for each fragment occur at the same pace, where individually labeled fluorescent nucleotides are being incorporated and each with a different dye. So once it's incorporated, it will be washed off, okay, and there will be image. So once they capture the image, they clean off the dye, okay, and then they're followed by the second reaction, second cycle, and each individual nucleotide with the label will be incorporated based on the template strand. So the cycle will continue until, okay, all of the template DNA is being sequenced. So if you look at the example of results that they will get, for example, there are two locations being imaged here, one location and the second location. If you see that this is the green in color for the first cycle, it means that this is a C. This is also green in color, this is also a C. This is for the first cycle. In the second cycle, this is blue in color, which means that it is an A. It is still green in color, which means that it is still a C. So if let's say there's 1, 2, 3, 4, 5, 6 cycles, you will have 6 nucleotide reads. 1, 2, 3, 4, 5, 6. So the first cycle is green, so it's a C. Second cycle is an A, it's A here. Red color is a T, green color is C. Yellow is a G, and the last one, red, is a T. So the bottom one, it is green throughout, which means it is all C. So for this type of platform, Illumina Solexa platform, they are using bridge PCR and they are using the cyclic reversible termination method. For this kind of platform, their error type is more prone to substitution, where the nucleotide incorporated may not be the correct nucleotide. So it should be a C, but a G is incorporated. So in this case, this platform, their error type is more prone to substitution rather than insertions or deletions, because all of them, they are going on the same pace or same rate as compared to the 454 platform. So for the ABI solid platform, they are using a different approach of sequencing. They are using sequencing by ligation approach. And similar to 454, they are using emulsion PCR, where the beads are being attached with multiple DNA fragments. And the beads, instead of depositing into a multiple well plate, they are being anchored on a solid planar substrate on a glass slide. So what do they do is they use a dinucleotide sequencing probe. So each of the probe with 4 different labels has a specific nucleotide. For example, let's say you see a blue dinucleotide probe with a blue label. It can be AA, A, it can be CC, it can be GG, or TT. So a blue colored label dinucleotide probe, it has 4 possibilities. It can be AA, CC, GG, and TT. So how do they do the sequencing here is sequencing by ligation, which is different from 454 or the Solexa approach. So in this case, you can see that you will have 5 primers. With the primer N, okay, so you incorporated the first one. You have the template, so the first one is incorporated and ligated, incorporated and ligated. So this ligation cycle will continue for the template sequence. So for the next cycle, you will need to have a primer with N-1, which means that you have a shorter primer with 1 nucleotide cut in. So from here, you would interrogate another nucleotide here. So again, the ligation cycle will continue. And these primer cycles are reset for 5 nucleotides being reduced. 1, 2, 3, 4. So there are 5 cycles with the original primer together. So at any one point, for example, if you look at this point, at any one point it will be interrogated twice. That's why they are called interrogation by dinucleotides. At any one point, it will be interrogated twice. So if let's say you get the color sequence, which is blue, yellow, red, with this sequence, and you know that with the primer sequence, you know that the first nucleotide is A. If let's say for blue, it can have 4 possibilities, right? AA, CC, GG, and TT, according to the probe set here. So if let's say you know that the first nucleotide is A, okay, if let's say you know that the first nucleotide is A, based on the primer sequence here, you know that this primer is A, the last nucleotide is A. So you would know that this blue is from AA instead of these 4 possibilities, instead of these 3. So it would be AA. So the next nucleotide A, if let's say you see a yellow color, yellow represents AG, okay, yellow represents AG, CT, GA, or TC. So they have 4 possibilities here. So if you know that the first nucleotide is AA, so again this would be sharing the same nucleotide. Okay, so it means that the second nucleotide, which is interrogated twice, it would be an A. The third nucleotide, it would be a G. The fourth one would be C. So with this, you would know, based on the color coding, the sequence of the template. So it would be A, the original, the first one, followed by A, G, C, A, G, T, C. So this would be the output sequence. So pollinator is a related sequence, related system similar to the solid platform. And the cost of this instrument, however, is substantially lower than that of the other second generation sequencing instruments. So what are the limitations for this is that emulsion PCR can sometimes be cumbersome and technically challenging, and they only have very short read lengths, only about 13 base pair. However, the advantage is that they are able to achieve extremely high data densities. You can obtain a lot of data at the same time. For the Heliscope platform, it is the first single molecular sequencing technology. So how does it work for this single molecule real-time DNA sequencing? So again, they are using the anchoring method, okay? So in this case, they'll incorporate one nucleotide at a time. So if let's say the first round they're incorporated labeled C molecules. So if let's say only this template has C, so it will be incorporated while the rest would not have any incorporation. So for this kind of platform, it is incorporation versus non-incorporation. So you wash off and you have a signal here, and you proceed to the next cycle where you incorporate it with another labeled nucleotide. So for example, this round is labeled C, the next round is labeled G. So again, depending on whether the template has a C, a labeled G would be incorporated, and then it will be washed off and the cycles will be repeated. So for this, you will not have multiple color images, but instead you will have one color image. So for example, again, you will have two location here, location 1 and 2. If let's say you see that there is a signal here, it means that this is incorporated. So it's C, T, A, G for the top one. C, T, A, G, T, G. C, T, A, G, T, G for the top location. For the bottom one, it would be C, A, G, C, T, A. So it will be a one color image instead of the four color images like the previous platform.